And we're going to get those chromosomes begin to unwind a little bit, form chromatin again, and then we get new nuclei created around each set of chromosomes. The mitotic spindle, of course, detaches. It has to if that nucleus is going to read here. And so you just have two sad little centrioles that are floating around, no longer attached to anything. The nucleus reforms, chromosomes begin to unwind. The nucleolus is going to reform in each of those daughter nuclei. And then what's going to happen is the next stage of the cell cycle, which is again not a stage of mitosis, but that's cytokinesis. So the cells will actually begin to divide. Now this happens a little bit differently in animal and plant cells. In animal cells, what happens is something called a cleavage furrow is created. So in cytokinesis, do you guys mind if I erase that chromosome?
Mm-hmm.
We start off with the cells that look something like this, right? This is the end of telophase. And we have two nuclei on either side of this cell. What's going to happen is the little actin filaments that completely surround the inside of our cells that facilitate amoeboid movement and other kinds of things will begin constricting along the equator of the cell, right along here. They're going to begin constricting that cell so that you get the cell kind of growing in on itself like this. And that's called a cleavage furrow. And then those actin filaments are just going to continue to constrict until they completely pinch off in the middle like that, and you get two independent cells. This reminds me a lot of how my mom used to make cinnamon rolls. So when she'd make cinnamon rolls, what she'd do is she'd get a whole bunch of bread dough, right? She'd roll it out really thin, and she'd smother it with butter, put all of the yummy goodness inside, this brown sugar and cinnamon stuff, right? And then she'd roll it up, and there would be a log of pre-cinnamon rolls. And then what she'd do is she'd take a string, instead of using, I don't know why she didn't use a knife, but she didn't. She would take a string, and she would wrap a string around this log of cinnamon rolls, and then just pull the string tight. And that loop would pinch off a cinnamon roll. And that's exactly what's happening right here. We have these strings that are completely surrounding the internal circumference of our cells. And then they just tighten up, and it just pinches the cell off into two. Plants are a little bit different though, right? They can't do that because they have a cellulose cell wall. And so instead what they do is inside their cells, so they have these rigid cell walls. And those rigid cell walls on the inside have plasma membrane like this. At the end of mitosis, there would be two nuclei. And so what they'll do is they'll begin laying down vesicles of new cell wall across the internal meridian of that cell, like this. And eventually, you'll get enough of a deposition of those vesicles that that cell wall will become contiguous like that, and then new plasma membrane will grow on either side to create two new cells. And I have a diagram of that. So this is what that cleavage furrow looks like, and you can see those actin filaments have actually begun pulling that plasma membrane in, and eventually it's just going to pinch off. And then here what you see is a cell wall down here, and a cell wall up here, and these vesicles being laid down in between two daughter nuclei. And eventually those vesicles will all become contiguous, and a new cell wall will form. Yeah,
Do
Carly.
plant cells look like a bunch of bricks, because they just are literally building a square after each other?
Yep, they do look like a bunch of bricks. Other questions? Okay. So I'll leave you with this slide, but I want to talk about apoptosis really quickly. So there is a process inside our bodies, particularly important in embryonic development and the control of cancers, called apoptosis, and it's called cell suicide. And what happens is cells, whether as a product of embryonic development or as a product of damage, can cause themselves to die. That process is called apoptosis. So in the development of a fetus, that's important because embryonically we have some structures that as adults we don't have, right? Like at some point, all of us had webbed fingers and toes in embryo. All of us had a tail. All of us had some kind of gill slit sort of structures. But as adults, we don't carry those, right? So what needs to happen is, at some specific time in embryonic development, that webbing between our fingers and our toes needs to be dissolved. And that dissolution occurs through apoptosis. Cells receive a signal that it's time to die. And they die, and we get independent digits instead of webbed digits, and the loss of a tail. Apoptosis, however, is also really, really important to control odd cells that are cancerous or precancerous. One of the interesting phenomena associated with cancerous cells is that they have an odd number of chromosomes. They can have more than 46 chromosomes, or they can have lots fewer than 46 chromosomes. And we inherently have some regulatory machinery inside our bodies that are able to detect cell anomalies like that. And our initial reaction is to try and repair it. If it's irreparable, however, then that cell should go through apoptosis and it should die. Cancer develops, however, when cells are able to sneak past that regulatory process. And we'll talk about how it is the cell cycle is regulated next time. So I will see you guys tomorrow in laboratory, and then I will see you again the Monday before Thanksgiving.